Our first speaker is Dr. Gerry Curatola. He is an internationally recognized restorative and integrative dentist, researcher, author, and clinician. He is Clinical Associate Professor in Cariology and Comprehensive Care at New York University College of Dentistry, where he founded the Curatola Wing for Clinical Research at New York University. He serves as the oral health expert to the Dr. Oz Show, ABC, NBC, and Fox television networks. He is co-founder and chairman of the CS Bioscience and founder of Rejuvenation Dentistry. Listed among America's top dentists and the leading physicians of the world, Dr. Curatola has authored numerous articles on dentistry and wellness and serves on the editorial board of the Journal of Dentistry, Oral Disorders and Therapies, the International Journal of Dental Science, and Globe Beauty Magazine. Please give a warm Academy welcome to Dr. Gerry Curatola.
Thank you. How's everybody doing? Woo! Oh, come on. I had a great night last night. My wife and I, we had this big bottle of wine, and after about 2 glasses each, I said, you know, honey, I can't believe we made 31 years. 31. Anybody married over 31 years here? We're like dinosaurs. So, and she just stayed quiet. She had a 3rd glass of wine, and then it was she held up her glass and she said, I love you so much. And I said, oh, honey, is that you talking or the wine talking? And she said, honey, that's me talking to the wine. So, really sweet, really, really sweet. Really sweet. Now, we had a discussion a little while back, and she said, you know, honey, if I died now, you know, she said, would you still remarry? And I said, well, you know, I know you wouldn't want me to be lonely, so I guess I would. And she said, well, would you give the new wife my diamond ring? And I said, well, honey, you know, you can't take these things with you, right? I said, so you wouldn't want me to go out and spend all that money, and yeah, I'd probably give her your ring. She said, would you give her my fur coats? And I said, well, yeah, they're just going to lay in the closet. I'd give her the fur coats. She said, would you give her those new Titleist clubs, the titanium clubs you got me last spring? And I said, well, no. She said, you give her my diamond ring and the fur coats, and you won't give her my golf clubs? And I said, no, honey, she's a lefty. Okay. All right, we're going to get started. I have a lot of things to share with you that are going to really change your understanding of what we've accepted for so long. And it's radically, you know, this whole microbiome, you're in for a treat this morning because my very dear friend and a brilliant naturopath and great researcher, Dr. Jack Tips from Austin, Texas, is going to be here right after me. And we kind of are a continuum because he's an expert on the gut microbiome, and I've actually spent 17 years researching the oral microbiome, and a lot of unbelievable things have been discovered in the last 10 years that's radically changed our understanding of disease. So let's see if I can. From when I came out of dental school, I really didn't want to be a barber dentist. I never wanted to be a drill 'em, fill 'em, bill 'em, or if you use mercury, I guess kill 'em, dentist. And I wanted to be probably the polar opposite of Dr. Bill over here. My favorite line that he has is right over here where he said, he's been using this same forceps for 50 years, still works, just not as pretty as it used to be. You know, so this is your classic barber dentist. I want to talk to you about being a physician dentist, a physician of the mouth. And Thomas Edison in 1902 had this. He was a lot better looking back then than when you see him with the white beard and you know, when he did the light bulb. But in 1902, he was saying that the doctor of the future is not going to give you medicine, but interest his patients in the care of the human frame, diet, and the cause and prevention of disease. And this guy is my hero. He gets way too little recognition. He's the Charles Darwin of nutrition. He is founded the research arm, or eventually became the research arm of the American Dental Association. But he was so far ahead of his time. This book, which he wrote in 1939, was sort of a culmination of his work. But what was really interesting about it is he was making the oral systemic connection back in 1917. This was an address to the St. Louis Dental Society and a branch of the American Dental, a very young American Dental Association, where he was talking about the relation of mouth infection to systemic disease, and then again, dental infections, related degenerative diseases. The thing that Weston Price did, which I don't see the slide, is he made the connection about 100 years before the U.S. Surgeon General that not only does oral disease affect general health and well-being, and not only does it affect you throughout your life, and not only is it this oral systemic linked to other generations, but here's the most amazing part. It affects craniofacial development, not only in future generations. That's called epigenetic expression. So what we're really finding is that this whole world of epigenetics is really extraordinary, where we actually change disease, damages genes that affects genetic expression in future generations. And so we are all well aware of the statistics, and some of them are even higher than here, but everything from Alzheimer's to colorectal cancer, from one end to the other, has a link to oral disease. And we're going to talk about this a little bit, because the mechanisms are not just bacteria from the mouth float around the body and do this infection. As a matter of fact, it's really less of an infectious disease than an inflammatory disease. And so the challenge we've had has been that 85% of adults still, in some areas, I mean, I visited with the Minister of Health of the government of India, and I sat there, and the man had rampant periodontal disease, and this is supposedly the leader of #2 under the Prime Minister in India. Met with him at the dental school in Hyderabad, India, and there you have, you have stages of periodontal disease over 90%, 95%, even the ministers of government. I want to focus on the third bullet, and I'm going to tell you a little why that these past approaches, the scorched earth policy we've had, is not only ineffectual but harmful. It is not only ineffectual but harmful. So whether you're using tea tree oil or triclosan, I'm going to talk about some of that, and I'm actually going to talk a little bit about my views on ozone therapy and where it has an application and where it most certainly does not. So in 2004, Time Magazine's cover said that inflammation is really the secret kill. It's these fires burning within. Now, inflammation is a natural body process. Acute inflammation, you get a splinter in your finger, ouch, you get out the offending foreign body, and you go through this inflammatory process that resolves and heals. What happens with chronic, low-grade inflammation, which gum disease is #1 source, it's a silent alarm bell going off. And there are various mechanisms, 3, where inflammation causes a chronic low-grade inflammation causes a cascade of events. And this cascade of events is really important to understand. Another great book I read, this book I read around 2002, 2003, was by Paul Ewald, called Playtime, and it really talks about how stealth, this is the whole idea of stealth infections, right? And we know patients come in and they're brushing and they see red on their brush and they think, oh, that's just natural. They were brushing too hard. And I always tell them, well, if you saw blood on your hairbrush, would you think you were brushing your hair too hard? No, that's not normal. That's a sign of inflammation, one of the earliest signs of inflammation. So we know, and Time Magazine said that gum disease is probably one of the body's #1 sources of chronic low-grade inflammation. It's the most ubiquitous source of chronic low-grade inflammation. And that inflammation has an effect throughout the body, and there's a number of mechanisms that we looked at. We, for many, many years, looked at the fact that inflammation triggers the liver to release C-reactive proteins, and they became a bigger inflammatory marker than high bad cholesterol levels in the body. By the way, we know cholesterol is not bad. Cholesterol has a reparative function. Your brain needs cholesterol. We have people taking statin drugs, dropping their cholesterol below 100, some of them, and it's deadly because cholesterol is not the culprit. You know, you are looking at the symptom and not the cause. It's inflammation that's the cause. Cholesterol is a sign of things going on, but you don't want to hammer cholesterol. You want to hammer the inflammation. So a study in the Journal of Periodontology reported that inflammatory effects from periodontal disease, a chronic bacterial infection of the gums, causes oral bacterial byproducts to enter the bloodstream and trigger the liver to release proteins such as CRP. And that has inflammatory effects on the whole circulatory system. This is one mechanism. It's gone way beyond this now, because we're looking, and this was back in the 90s, late 90s, that a lot of these concepts were being promulgated, because we knew that it was beyond a bacteria in your gum setting up shop in an artery around your heart. We knew it was way beyond that, and we knew that there were other mechanisms going on. And then we started to really, in about 2000, we launched, 2002 rather, we launched the Human Microbiome Project. And that sort of redefined what it meant to be human. And it's really very interesting. I mean, there are even theories right now. So there are 10 times the number of bacteria to every human cell. And the genetic information from those bacteria is 100 times to our own human cell. The idea of thinking of bacteria as bugs and invaders, right now, it's pretty well accepted that we were made from them. So whether you are an evolutionist or a creationist, I just came across the Hebrew Torah in Genesis, where God says he took from the fertile soil. A lot of the regular Bible versions are he took from the dust of the earth and he breathed into it, and that's how man was made. So, you know, he created the earth, created the microbes and everything else, and then he breathed his life into it. That's his spirit in our mortal body. But I actually found the Hebrew version really interesting, because it says, and God took from the fertile soil, which was all microbes, and he breathed into it. And there's a lot of theories right now that our human cells, which are grossly outnumbered, and by the way, that number has gone from 10 times more microbes to now 3 times more microbes. We just know it's a heck of a lot more microbes than human cells. That's what we are standing here or sitting here right now. And so what we know is that in the mitochondria of in the nucleus of our cells is a little energy factory called the mitochondria. The mitochondria are very similar in size, shape, dimension, and function like a unicellular organism, such as a bacterium. So there's this bacterial origin of the mitochondria that's out there right now as well. So like literally we are 100% made of microbes. And that is a unique, very different understanding than we've had, you know, especially when you think about the Cold War of microbe and antibiotics that we've existed in from the time that penicillin was invented. So the Human Microbiome Project kicked it off. We knew a little bit more before this. This was when the first formal study became a huge avenue for research. And this is really, we know that the microbiome has a huge impact on our health, okay? It's our ability to digest food and more. And I'm here today to tell you that trying to kill the oral microbiome, because we think it's bad, and believe me, microbiomes, if they're out of balance, can wreak havoc in everything from an oil barge to a food factory, you know, that gets a biofilm in one of their water supply pipes. And I was just meeting with a manufacturer for an oral care line that I've developed that is, he was telling me about problems he had with biofilms and how they have to shut the entire FDA facility down just to get one little patch of a biofilm that's stuck in a pipe somewhere. So I want you to understand that these microbiomes have paid a high evolutionary price to be there. Second, I want you to understand that these microbiomes keep us alive, especially in the mouth. And I also am going to address the fact that the oral microbiome, which we work in every day, the oral microbiome is probably one of the most unique of the entire human body, and it's very different and a cousin to the gut microbiome. But probiotics do not work in the mouth the way they can be mildly effective in the gut if you get past the hydrochloric acid of the stomach and that whole microbiome of the stomach, which is about this thick, and it's filled with H. pylori and all. Okay? So we understand that, and I'll explain why probiotics don't work in the mouth, and that's the same reason why conbiotics don't work in the mouth. Because the oral microbiome protects you, keeps you alive, aids in digestion, and it's an essential component of your salivary immune system. So this whole microbiome project reminded me of when I was a kid and I'd sit in front of the TV on Sunday and watch Jacques Cousteau, the amazing undersea world of Jacques Cousteau at the Sargasso Sea. So Jacques Cousteau did a lot of great things. First of all, he was the first time that we could actually see underwater. We really, you know, the whole sport of scuba was invented by Jacques Cousteau and a partner in the early 1950s. He invented the aqualung shortly after World War II. So this amazing world of biodiversity was made possible because of new initiatives and new tools by an explorer, a marine explorer named Jacques Cousteau. And I said, wow, what a parallel to our amazing biodiverse universe living inside of us, in and on our bodies. 5 major microbiomes that we have identified: oral microbiome, gut microbiome, respiratory microbiome, genitourinary microbiome, skin microbiome. And there are more subcommunities. So we saw the human oral microbiome is truly the gateway to systemic wellness. It's essential for life. It's essential for protection against deadly environmental pathogens. It's an essential component of mammalian digestion, and an essential component of the salivary immune system. So this whole idea of plaque, what is plaque? Okay, you got your perio probe on the right there, and I'm here to tell you that what's being scraped off the teeth, which we traditionally would put on a slide and show patients bugs swimming around, that is an unhealthy expression of a healthy human oral microbiome, because a healthy human oral microbiome is a thin, clear, odorless film. And it's there whether you see it or you don't. And you're dealing with about 600 to 700 different species of organisms that all have a uniqueness so that your oral microbiome in your mouth is different than the person sitting next to you right here. It's as unique as your thumbprint. And so how did we learn about all this? And this is the big thing. Our aqua lung that Jacques Cousteau invented, our ability to see underwater, our ability to learn all these things came about because we started using some very sophisticated diagnostic tools. And there were three major ones in the study of the human microbiome, especially the oral microbiome. The first is, atomic force microscopy. We are able to look in vivo at the human oral microbiome without disturbing it. So it's no more taking it out, smearing it on a glass, and looking at a bunch of pathogens spewing around, because those pathogens are really I'll explain, they're a brief transport. Looking at the pathogens is looking at the icing on the cake. It's not looking at the cake. Okay, pathogens spew off, and these same bacteria that were living very peacefully go wild because of environmental conditions, which that's where the focus needs to be. So atomic force microscopy gives us this topographic map, and you can see different hills and valleys, and that's how we started to study when I show you some illustrations of the oral microbiome, you'll see these different strata and streamers and tunnels and hills and valleys like you see on this map on the right. And then we had confocal laser scanning microscopy, and this gave us the ability to do optical sectioning. So we're able to look at different layers, and we could see a basal layer closest to the oral tissue, teeth and gums. And then we could look at layers above that, because the top layer in an unhealthy human oral microbiome, that top layer generally is loaded with the badass bugs. Spirochetes, Fusobacterium, they're laying on the top. And they have smushed out this beautiful aerobic layer. Okay? And that's very, very interesting. These are the tools that have completely changed our understanding of the human microbiome. The final one I wanted to tell you about was these fluorescent probes. And this is like live staining. So we're able to label or stain living cells and protein molecules with light absorption and emission, again, without disturbing their native environment. And what we found is that bacteria in the mouth exist in two different states. And one of them I mentioned, this free-floating or planktonic existence. That's what we have traditionally been smearing on a slide and putting under a microscope to show and gross out your patients of all this stuff that's swimming around that we have to kill. This is the stuff we have to kill. And that's our familiar background. But now we know that this planktonic, that thick, sticky, smelly plaque is a oral microbiome that's out of balance. And I'll explain how that happens. But bacteria in your mouth that keep you alive, they actually live in a more common existence like we do. They live in communities, and it's kind of like, I remember when I was about 10 years old, my dad took me to the part of Brooklyn where he grew up. And it used to be this beautiful part of Brooklyn, New York, that had these brownstones next door to each other, and everybody had flowers on the steps, and everything was so well kept. The streets were clean, the garbage was picked up, the houses were being painted. Yeah, the milkman was delivering the milk bottles, and and all this stuff that was going on in a normal functioning community. And then you had environmental conditions, economic conditions, that changed that, and economies changed. Things happened, and the community went out of balance. And the next thing you know, you had burned out brownstones, cars on fire, people getting looted, gangs in the street, and that's what happens in our mouths. When you put things, another analogy I used is you take a bunch of high school girls that wear uniforms, and they're on a, and they're all really nice. You take them to Cancun on spring break, and you give them a lot of tequila, and you have girls gone wild. Okay, so that's another analogy I use. But bacteria can do the same. The same well-behaved, well-functioning, beneficial bacteria they flip a pathogenic switch. And they, by the way, they vote. Everybody here knows they vote. It's called quorum sensing. Okay, so they get together when they're under attack. Pesticide. They're under attack. Basically, they hunker down. They even secrete a protein shell. We discovered that because we took plaque off the tusk of a mastodon frozen in a glacier. We put it on a petri dish. We took just a scrape off tusk of a mastodon. They put it on a petri dish. They put it at body temperature, and they saw these microbes unflourish after being frozen in a glacier. And they called those bacteria persisters. And they called them persisters because they secrete a protein shell. So we are not going to nuke bacteria, and we're not going to get rid of and be able to kill plaque and destroy the oral microbiome, and thank heavens we didn't, because we would have unleashed ecological Armageddon. And now the understanding we have is to promote balance or homeostasis. It makes it very unfavorable for Fusobacterium nucleatum, which is probably one of the biggest thugs, gram-negative bacteria. It's a gram-negative anaerobic bacteria. It is a badass bug. It actually carries other bacteria on its back to the brain and to other places that have been linked to amyloid plaques and all kinds of other things, which I'll show you some research with. But Fusobacterium nucleatum live in a beautiful biofilm. They live there. They're kept in check. They just kind of do their thing. But they become opportunistic when the environment is out of balance. So what we don't want to do is say, there's a spirochete, there's a fuso. These are brief transports. You're going to see spirochetes in the mouth, everybody. We're never going to sterilize the mouth of spirochetes. You're never going to sterilize the mouth of Fusobacterium. We're never going to be able to, and you do not want to. What you want to promote is balance, not nuclear war. So it's microbiome imbalances or a lack of homeostasis, not specific bacteria that are causative agent. And I said, when there are adverse conditions for these bacteria to survive, they kind of get together, exchange genetic information, quorum sensing, and they flip a pathogenic switch. And then all hell breaks loose. And we're going to look at that because I think Phil yesterday was talking about a lot of research done. A lot of the research on the oral microbiome, great research, came out of University of Montana. Any Montanans here? No Montanans. All right. In Bozeman, and the Bozeman campus. And but this is January 2015, okay? The Journal of Alzheimer's Disease, the Microbiome Disease, reviewing the links between oral microbiome, aging, and Alzheimer's disease. Wow, we knew it was there. Now we really have a lot of mechanisms. And we're looking at this, again, inflammatory response, where astrocytes are recruited to these inflammatory areas, and they become hypertrophic, and then there's a whole cascade of events that goes on. And a number of them are this whole idea of immunosenescence. Immunosenescence is where your immune system becomes senile. In other words, your immune system doesn't really work as effectively as it should. And it happens because of a hammering from an increased bacterial load. And there's also, we have antibody responses getting screwed up as well. This whole idea of a rise in circulating pro-inflammatory cytokines, again, related to inflammation. this integrity of the blood-brain barrier getting against increasing bacterial load. So, you know, we have the blood-brain barrier keeping bacteria from getting passed into brain tissue, into cortical tissue, when instead what happens is that blood-brain barrier, the integrity of that gets beat up, and then the progress of this is really, really big. And we're actually looking at once someone, you know, it takes about 20 years before Alzheimer's symptoms to surface. So it's a scary thought, but it can be subclinical, or what they say preclinical, until preclinical diagnosis 20 years before. But we're seeing this whole cascade that happens between the oral microbiome. So do we sterilize the mouth, or do we promote oral microbiome homeostasis? And there's also this bidirectional link. And again, this metastatic spread of gram-negative bacteria, F. nucleatum. Fusobacterium nucleatum live in the mouth. They are a commensal bug. It's a community bug. And, but you'll see what happens when there's a lack of homeostasis. So these are all the bidirectional links, including the fact that periodontal disease increases your risk of stroke. So you have cerebrovascular injury, and this is all the link between periodontal disease and Alzheimer's disease. And this is becoming an increasing concern because we're finding that gum disease may be one of the biggest causative links between the mouth. You know, they were looking at gut bacteria, but now oral bacteria, oral pathogens. Another big area is looking at polymorphisms in the interleukin-1 gene. The interleukin-1 gene is being focused on a lot, promoting an increased inflammatory response. So let's look at how we go from the left to the right. So the left over here is acquired pellicle. This is an atrophic biofilm. So every time we blast the mouth with an antimicrobial, so if we used triclosan, or we use chlorhexidine, or we use tea tree oil, or we just ozone the hell out of the mouth, you're really going to create this atrophic area. This is the sweet spot. This is a thin, intelligent, semi-permeable membrane. This is an oral biofilm, an oral microbiome. By the way, we changed the word from the oral biofilm to the oral microbiome. This scientist, Josh Lederberg, recoined the term biofilm to microbiome in 2002. So we've been using this fancy microbiome word since around that. But before that, we called it the oral biofilm. So how do we go from this great area here where there's this aerobic predominance of aerobic bacteria, healthy tissue, thin odorless film, your teeth don't stain, your teeth are whiter, your breath is fresher, to this over here with this whole inflammatory response in the soft tissue and decay in the hard tissue. So how do we go? And I'm gonna have you focus on this chart. This is one of my favorites. So we want to be in the center of the target. To the left is the atrophic biofilm. That's what you saw on the left over here. And to the right, you have this hypertrophic. And below you see mostly soft tissue, oxidative reduction reactions in the soft tissue in the mouth. And above you have acid-base reactions, mostly hard tissue. And if you go into this corner over here, and I heard Phil yesterday talking about ozone is pretty good for reducing sensitivity, and I think that's great, because the way you reduce sensitivity is you block up those tubules, and one of the ways, if you have a healthy oral microbiome, you are not going to be over here. You're going to be over here. If you have a hypertrophic biofilm, that's where you have all that gunk on your teeth and you get tooth decay and gum disease. Another area with atrophic biofilm is ulcers and burning mouth. Did anyone here ever have patients report ulcers or burning mouth from Colgate Total toothpaste? Okay, my hygienist, she just polls people and says, what are you using? Oh, I'm using Colgate Total. So you get that atrophic biofilm, and that's where you get these ulcers and burning mouth. Because you're really nuking the entire, you're just really stripping it. So you really don't want to be atrophic and you don't want to be hypertrophic. So let's talk about it. It is an intelligent semi-permeable membrane. Again, a lot of work was done in the UK and in Montana. Very, very important to understand the function of this oral microbiome and that this beneficial biofilm. Not let's kill the bugs, because every bug is bad and you got to kill them. It's like, you know, it's probably the worst type of prejudice we can have is against our own bodies. Every bug is bad, kill it. The oral microbiome exchanges with the structures on its sits. So, and it's basically, how it does this is dependent on the environment. So if you are eating a high carbohydrate diet, high sugar, you are going to cause an unfavorable environment, causing the overgrowth of acidophilic bacteria, and you have an imbalance in this microbiome. Alcohol, chemicals, salivary interruption, cancer patients. I mean, there's all kinds of things. So the microbiome, the oral microbiome, is a storehouse of minerals and ions, keeps enamel hard and pH neutral. And the organisms, if the organisms are, you know, so if you have an unfavorable environment and they demand free calcium or phosphate, you get a deficit of these minerals or ions, resulting in a low pH and demineralization. So this concept that this is a living, breathing storehouse, or in deficit, goes beyond commonly, you know, the calcium and the phosphate and ions. But what we know and what we're looking at now, the hottest area of research, is this area of balancing molecular and ionic oxygen. And molecular oxygen is really what we want. And when you have this thin, odorless film in the middle there, full of aerobic bugs, they help to transport molecular oxygen to the gum tissue, stay pink, stippled, nice, hard, don't bleed, and you get this exchange. But in an unbalanced microbiome, that's where you have this charged oxygen, it's ionic oxygen, and the bacteria will actually use ionic oxygen to poke holes in the soft tissue. And that's when you see gingivitis. So that's where the unhealthy radical or ionic oxygen happens when you have the biofilm being out of balance. So it's all about promoting balance. The oral microbiome is basically water, salivary proteins, cervicular fluid, immune complexes, minerals, and about 700 species. 6 to 10 billion, this goes up and down, 7 to 10 billion organisms, and you have everything in there. You have protozoa, you have aerobes, anaerobes, fungi, you name it, are in there, and they live in this community. So let's take a closer look at this community. It's this aerobic layer that is the most beneficial, but it also is this aerobic layer that is the most perishable. So the another thing is, how does this microbiome work with saliva? And they work together, passing food through. So the salivary mucoproteins are like the oil, and the microbiome is like the ball bearings that help the food slide down, and reducing friction of a bolus of food that goes by. And because of those three tools, we really see this very complex community here. And you have these cell clusters and streamers and channels and all these different mixed species. And so I talked a little bit about quorum sensing, because they can sense the own number of their own species through a process called quorum sensing. Great TED Talk. Great TED Talk, someone I'm going to be interviewing on a TV show that's going to be launching. Bonnie Bassler, microbiologist from Princeton University, did one of the best TED Talks on the microbiome, and it's called How Bacteria Talk to Each Other, or How Bacteria Communicate. She does a great job. How Bacteria Communicate, Bonnie Bassler. So this is a basic, healthy, regular, stable structure that goes through just what I said in a normal community. Nutrient collection, waste removal, collective survival. They can modulate their positioning so they can challenge a threat from a topically applied antimicrobial. And they can actually, they're a persister bacteria that actually kind of secrete a protein shell, hunker down, and then open up again. So when the microbiome goes from this aerobic state, these early colonizers get matted down and convert from aerobes to facultative anaerobes. So that's what you see. So this beautiful yellow and green layer becomes all gray here, and these guys start packing on top. The Fusobacterium and the spirochetes, and you have these opportunistic organisms pile up and destroy the architecture of the basal layer. So really, this beautiful, thin, odorless, protective, beneficial biofilm that's an essential part of mammalian digestion gets completely beat up by this over here. By the way, this is what Weston Price was looking at too, in so-called civilized diets. We were creating this. But in healthy primitive cultures, we didn't see, or he did not observe, the plaque and decay and periodontal problems and disease that we saw, or that he observed from civilized diets. He even saw that their posture was different and everything. I mean, so many brilliant discoveries, anthropological discoveries he made in studying these cultures around the world. So this just came out also. This is January 2015. Fusobacterium nucleatum, a commensal turn pathogen. So how does it go? And it says here, Fusobacterium nucleatum is an anaerobic oral commensal and a periodontal pathogen associated with a wide spectrum of human diseases. From preterm birth, stillbirth, they're going into here, colorectal cancer, inflammatory bowel disease, appendicitis infection, Lemierre's, Alzheimer's. So where do we go? How does Fusobacterium go from being a commensal to a pathogen? And it's all about what we're doing to deal with it. All we do when we take this approach, okay, let's kill it, whether you use an iodine, or remember we used to wash with betadine liquid. I remember when I went into my residency, I was using betadine, and scrubbing up with that, and then in a matter of weeks I had weird fungal infections and nail beds that looked like crap, and realized I was destroying my skin microbiome. And we do that in the mouth worse than ever. And I can only say, by the grace of God, we have not gone too far, and that the mouth really rebounds. You know what happens when you promote oral microbiome homeostasis? The mouth returns to a state of health faster than you can believe. I mean, I've observed gum stop bleeding in 6 minutes. So we have got to stop this, and don't kid ourselves. I'm going to show you a little bit about other things that are disruptive to the oral microbiome, which we thought was good. It's anti-plaque. Xylitol is great, isn't it? It's all natural. It's all natural, right? Isn't it natural?
Oh, it's made by a chemical process of hydrogenation using heavy nickel, rainy nickel, as the catalyst, so that it makes plaque slippery on teeth, so plaque doesn't stick and it disrupts the plaque. Oh, but I get really bad intestinal gas, and oh yeah, and I was in the bathroom for a while after chewing 3 pieces of spry gum, because it disturbs, it's not absorbed, it's not metabolized, it's a high-value chemical that we've been sold as natural. Yes, does xylitol occur in nature? Sure. Xylitol is in nature. Comes from birch bark, right? It's in nature. So is arsenic in apple pits. Okay, it doesn't, just because it's in nature doesn't mean it's good for you. Okay, there are deadly fruits and vegetables that we cannot eat. So the goal becomes driving this to homeostasis, a low film thickness. The goal is to get it to go from this to this. Do we get it to go to this by just wiping it out and starting again over here? And my answer to you is no, but. Okay, if you have a condition in the mouth where there is an active major periodontal infection, osteonecrosis, a major periodontal abscess where the body's overwhelmed, you're not going to go from this to this so quickly. And I am not saying here, and I'm not standing here to tell you that there isn't an application for an ozone therapy or it is an application for an antibiotic. That is not what I'm saying. What I'm saying as an oral care therapy, as an everyday therapy, you better not be brushing with ozone, and you better not be brushing with a tea tree oil toothpaste. Tea tree oil, by the way, is a very powerful oil. It is a terpene. It kills everything, even your pets. Okay, there were more tea tree oil poison control center emergencies than any other essential oil on the market last year. So we got to know what we're talking about to promote a rational, sane therapy for promoting oral health. The American Dental Association said yes, it is a microbiome, a biofilm-mediated disease. We are mediating the microbiome. Remember this chart, where you want to be. And what happens is we overreact when we see this hypertrophic, and we immediately go to the atrophic. And so we're looking at periodontitis. This is also a 2015 study that just came out in an immunological journal. Periodontitis is a dysbiotic inflammatory disease, and this review discusses the mechanisms of microbial immune subversion, tipping the balance from homeostasis. We're starting to see that word. I've been using that word for 8 to 10 years in oral or extraoral sites. Another great study, also not that long ago, the oral microbiome and health and disease and the mechanisms. And it says down here, in particular, although caries and periodontitis, I might as well show you the slide, although caries and periodontitis are clearly bacterial diseases, they are not infectious diseases in the classical sense, but they're a result of a complex interaction between commensal microbiota, host susceptibility, and environmental factors. That's really the mechanism. Not that these bad bugs set up shop in your mouth and all hell breaks loose. The same bacteria, these same pathogens, H. pylori bacteria live in your gut microbiome. Staph bacteria live on your skin. Staph aureus is on your skin right now. Pneumococcus bacteria are in your lungs right now. So how do you get bacterial pneumonia, staph infection on your skin, H. pylori? Oh, I have an H. pylori infection in my gut. Let's just kill all those H. pylori. They live there. They flip a pathogenic switch when they're under attack. So maybe you should think about laying off the fried food and reduce your stress level and and the amount of hydrochloric acid. By the way, what are all these Prilosec and everything? You know, right? I mean, we have statins just destroying people's cholesterol. And then you have these antacids and these proton inhibitors that are changing the microflora of the gut, because the acid in your stomach prevents some really bad pathogens from getting by and getting into your intestines. So we start to see an altered gut microflora, and that has very profound consequences. But I said something before. I said, oh, probiotics. We got to use probiotics in the mouth, right? Probiotics work in the mouth. If probiotics work in the mouth, then conbiotics, right? Well, what are conbiotics? Salmonella, E. coli. So why don't you get salmonella in the mouth, right? You get a salmonella infection in here. Just get a nice, bad cheeseburger, juicy, greasy cheeseburger. It's in the cheeseburger, gets past the mouth, somehow gets around the hydrochloric acid in the stomach, and it gets in the gut, and now you have a conbiotic, okay, making you very sick. But that salmonella didn't do anything in the mouth, because your oral microbiome is unique. It protects you from deadly viruses and bacteria in the environment around us. Probiotics were tried in the mouth as far back as the 1970s. They do not work. And if anybody tells you they do, they really do not understand the mechanism of the oral microbiome. So it's a nice idea to throw lactobacillus around, and they are mildly effective in the gut if you can get them enterically coated past the, obviously, the stomach. But if probiotics could work in the mouth, and I'm telling you, thank God probiotics don't work in the mouth, because if probiotics work in the mouth, conbiotics work in the mouth. And so we have a real change in assumptions here, right? This is the classic way we treated an unhealthy microbiome in the mouth. Kill the plaque. Let's just do the scorched earth policy and start over, try to grow good grass. But if anybody tries to grow a grass like the pretty picture on the Scott's bag, they know the first thing to come back are the weeds. Fusobacterium nucleatum, probably identified as one of the biggest potential pathogens to systemic disease all over the body, originating from the mouth, actually would be the first to set up shop here. Okay? They are pretty tough. It's like a mafia bug, so before it joins the mafia, it's in this big Italian family. They have this and then it gets a made man, and that's when it does havoc all over for its own survival. So my point to you this morning is you really got to think of your mouth as a healthy, robust organic garden. You got 700 different species. You got 6 to 10 billion organisms in here, okay? And this monolithic approach would round up. By the way, triclosan is our Roundup in the mouth. You know about glyphosate? Everybody here know about glyphosate? A bane on the human body, thanks to our friends at Monsanto. So I was this summer in Ravello, Italy, laying by the pool, and my wife sees me jump off the chair. I'm reading the International People magazine, and there's a full-page ad here with Kelly Ripa with her bleached teeth, and it says, eliminate 15 times more bacteria to improve the health of your mouth. Not only is that, and Colgate, by the way, paid for a lot of the research on the oral microbiome. They gave Bozeman a lot of money, the Bozeman campus in Montana. So the idea that nuking the bacteria makes you healthier is the exact opposite. So I found this to be very disturbing, that they're allowed to continue to propagate lies that the science has completely disproved. So I'm going to go very quickly. I've got about 15 minutes, and I'm going to go very quickly through a few things because I threw a few bombs out here because I've come to a number of these meetings, and I love this organization. I really do feel we are going to see some exciting things, and I do believe that by 2020, that's my prediction, we will see some of the embarrassments that we have propagated in the interest of advanced dentistry, promulgated in the interest of advanced dentistry, eradicated and ceased to be put in use. I think one of the things that I feel very strongly about is that the science basis that this organization takes, the respect for science that this organization takes, is an important part of our credibility as a group. And so I want to give you a little bit of the science. This is really preaching to the choir, and we know all about this was thanks to Dave Kennedy and all of his great work. I do not need to go through this. I always show patients, we're still using saccharin in there and all this jazz. I just want to get past this. Patients still don't, they're like, why is it poison warning? You know, this still goes on. Artificial dyes, this is not a natural color. Everybody's up on this, right? We put put propylene garbage bag particles because it was cheaper to color Crest toothpaste with propylene glycol pieces that my hygienist was finding in cervical margins. And there's a great hygienist who blew the lid off of this, and there was a piece on CNN. How many saw that? About the propylene garbage bags. Yeah, they were using, here it is right here, Crest Pro Health for me. It's over here, propylene, polyethylene, I'm sorry, polyethylene. Propylene glycol is also used, but polyethylene, yeah, plastic kitchen bag particles to color the toothpaste people are brushing with, right? And do not swallow. But that's what they were using to color the product. Plastic. But probably the most insidious ingredient in conventional toothpaste was what Colgate used to take the lead from Procter & Gamble's Crest in 1998 with a $100 million launch budget. Triclosan is a known pesticide. It does not biodegrade. It was a human health risk, an environmental risk. Back in the 60s, I believe the FDA banned it, and then they allowed it to go back in. Finally, we are seeing the EPA has petitioned the FDA. The FDA has finally relented, and we're pulling it out. But anyway, researchers at Virginia Tech also found that triclosan mixed with chlorine forms chloroform. That study came out. I couldn't believe it. I thought it was going to be huge news, and it got buried because just as my appearance on Toxic Fillings on Dr. Oz got butchered because of lawyers afraid of the American Dental Association, the whole safe mercury removal segment that they filmed 6 hours in my office was pulled. And instead we talked about, oh, eat chlorella and take chlorella and eat a lot of cilantro, you know, but probably the most important part was the part that this organization created a standard for, which is safe mercury removal. So same thing with Colgate toothpaste. You know, you have all these billions of advertising dollars that have been spent, and we have all of these studies that have come out. The real scary part about triclosan is not that it kills everything, but that it reduces your body's, reduces the ability for other antibiotic to work in life-threatening situations. So you have decreased susceptibility to triclosan. You have the creation of new superbugs and a decreased susceptibility to other antibiotics. They found in 3 out of 5 randomly selected high levels of triclosan in human breast milk. It's also found in the bile of fish exposed to municipal wastewater. This is an environmental disaster with triclosan. It wasn't just, Colgate toothpaste was the #1 user of this, but you had arid antibacterial underarm thing. It was one antibacterial everything. It is, so we have antimicrobial, the emergence of antimicrobial drug resistance. Study after study. This is a CDC publication. So we had decreased biodiversity. So you had, because of the gray water used for irrigation, basically we're sterilizing our soil. The proportion of microbial isolates resistant to antibiotics increased, while at the same time, overall diversity of the microbial community decreased. This is as much an environmental disaster, poison, toxin. We created our own love canals everywhere. And actually, triclosan converts to dioxin in riverbeds as well. So, finally, I think we are going to see this. I got the honor of, because I'm a major donor to New York University, I got to escort Ian Cook, the chairman of the board, president and chief executive officer, in the academic procession. He was being given a humanitarian award for donating to NYU's clinics, charitable clinics, humanitarian clinics in different countries. Colgate has been a supporter, and so they were giving him an award, but I got to stand next to him for a little bit, and I said, when are you going to take that triclosan crap out of your toothpaste? And literally, he looked at me like a deer in the headlights and said, we're working on it. And so they are working on it. That was the #1, that's, by the way, everyone, that's the #1 active ingredient in the #1 selling toothpaste in the world. That's the active. And this is what it caused. Minnesota's banned it. They already passed a law, but the FDA quickly jumped in, and I think 2015 we're going to see the removal of triclosan from these commercial products, so personal care products. And so it has an almost indefinite afterlife in human tissue. Swedish studies found triclosan in 60% of women's breast milk, even after years after they stopped using triclosan products or antibacterial products. It's a known endocrine disruptor. It's associated with antibiotic resistance. It forms carcinogens when exposed to chlorine and tap water. Human disorders. This was an interesting one too, that it affects cardiac and skeletal muscle weakness. They also saw this because fish were swimming slower upstream. So, I mean, it's literally a major disaster on so many levels. So I'm going to finish right now. I want to talk to you about natural toothpaste, right? All natural toothpaste is good. And, right, no artificial color, no artificial flavor. I'm just going to, this is, I've been on the bandwagon with it. If there's one mindset I'd like to change in this room, it's the idea that xylitol is good. By the way, does everyone know about 80 to 85% of xylitol is controlled by DuPont? Do you know that? And guess what? It's their subsidiary Danica in Europe. It's very expensive to make xylitol from birch trees because you'd have to take down a lot of these beautiful, pretty birch trees. The majority of it has been shown to be made from GMO corn cobs. GMO corn cobs are basically thrown away after you use the GMO corn, and xylitol is very, very, it's not metabolized, and it's not absorbed. It was mainly used in dentistry. Here's the big thing about xylitol. The manufacturers love to use it because it's a great sweetener. So I call xylitol the methadone of the sugar addicts, okay? Because we had aspartame, we had saccharin, right? We had saccharin because we were starting to get addicted to sugar in the 60s. So we came up with a sugar substitute for diabetics. We called it saccharin, till we started seeing it cause cancer in mice and all this other stuff. Then we came up with another great one called aspartame. It actually converts to formaldehyde in the brain under higher temperature, right? So aspartame, that's another good one, right? And we have osteoporosis and all kinds of other things that were aggravated by aspartame. And then we had sucralose. And sucralose, made from, I think it's a wood alcohol, it has almost an indefinite half-life in human tissue. So the sweetness stays inside you. And now we have the sugar alcohols. And so what do we know? We know that we are a sugar-addicted country, and we are going to try and come up with sugar alternatives. So let's come up with a new methadone for our sugar addicts. Just like we use methadone for heroin addicts, let's come up with a new methadone. And why is it the new margarine? It's a new margarine because this is how it's made, okay? This is the hydrogenation process for sugar alcohols. And by the way, the catalyst is raining nickel in most of it. So do not be lied to, and do not swallow the bullshit.
See, the problem is
it is an unnatural, unhealthy sweetener, but it is a good sweetener. And you end up with a high-value chemical over here. That's what they call this. This is the hydrogenation process.
Your inquisitive look.
It's more like a scientist.
And here's a couple other nice little tidbits. There's enough xylitol in 2 pieces of gum to kill a 100-gram rat.
Yeah, there it is.
In fact, according to lab tests, a 100-gram rat can be killed by 1.65 grams of xylitol about half the time. And by the way,
good to go.
I want you to look up the long-term safety data on xylitol. There is none. Okay? No long-term safety data. This has been shoved down the throat of so-called natural dentists, biologic dentists, healthy dentists. It disrupts plaque. It does a lot more than that. And let's look at the cousin, erythritol. Now, xylitol is poorly absorbed and poorly metabolized. So it's not absorbed and not metabolized. And that's why it ferments in the gut. That's why you get bloating and gas with any kind of ingested xylitol product. However, erythritol it contributes to metabolic acidosis because it gets absorbed, but it's not metabolized. So it leads to acid reflux, to increased risk of cancer of the larynx. These are the things that you have to look for. So another sugar alcohol. Again, naturally sweetened. I just got elected to the board of the Organic and Natural Health Association, a not-for-profit in Washington, D.C., that's going to go after all these claims. This is a chemical, everybody, and kills fruit flies. Who have a DNA pretty close to us. Yep, kills fruit flies. So they're looking at it as an insect repellent now, erythritol as an insect repellent.
Very good to meet you. We've talked on the phone a few times.
Tea tree oil, very volatile.
Way too long.
Okay, can be very toxic. I'm going to go very quickly there. Naturally sourced fluoride.
Are you a dentist?
Preaching to the choir again. Sodium bicarbonate is very abrasive, okay?
I don't know.
That abrasive.
5, 6, 7, 8 years or something.
So I was just interviewed by Dr. Mercola after he saw this. But here is Dr. Mercola doing a safe natural toothpaste. He has 1, 2, 3, 4 antimicrobials, some of them powerful antimicrobials. This thing will cause an atrophic biofilm in a heartbeat. This is not what you want to be doing. Very quickly, I'm going to finish right now and wrap up. I have developed, and just so you know, I'm a co-founder of this company, which was founded in the late 90s.
Are harmful for your eyes.
We started this as sort of a bioactive, biologically active, and a...
How cool.
Really, it was designed to feed your smile. I just was so sick of toothpaste. and came up with a nutritional homeopathic formula. We got 3 patents on. We found these things happen. We started doing studies. And in 4 days, we went from that to that. Our reduction in gingival inflammation was really big.
Dr. Curatola, he's a pretty interesting guy.
These were our...
I've already seen his presentation.
In 1 week, we had 72% reduction in bleeding, 46% reduction in this. I will present on this next time I am here, but I did want you to know one last thing. That's where you can reach me if you want. I have some exciting news. I'm going to present on, hopefully, be exhibiting because I am really, I've been frustrated for 31 years on the types of oral care products we've embraced in this profession, and there's got to be a sane alternative, and I'm trying to do that in the work that I've been committed to over the past 20 years of research in this. And so committed that I actually built a clinical research wing at NYU for translational research and in the development and introduction of oral care therapeutics. So I want to thank you all very much. Thank you for having me again. And I hope you enjoyed this.